So when's the midterm? Wednesday. So week from today, right?
Yes.
What that means is the material on the midterm will be today's lecture, Friday's lecture, Monday will be a review so I can try and catch up on the stuff that I did a really bad job of explaining the first time around, and then it'll be the midterm. Midterm is 50 multiple choice questions. You can have as long as you like. It shouldn't take you more than about half an hour. So that's, again, completely up to you. Those of you who are registered with the DRC, make sure you remind me beforehand, and I'll make sure the exam gets over there. Okay, question on the exam, yes? Yes, the large Scantron. I will remind you of that multiple times, don't worry. Yes?
So Wednesday or Monday's going to be a review, so can we
So the question here is the question about questions. Since Monday's going to be a review, can we come with lots of questions? Yes, that's a great idea. Again, what I'm going to do is I'm going to pull off slides. They'll be exactly the same slides that I've used before. And so if you've got extra questions above and beyond that, great. If anything, I was just talking to one of my colleagues over in the biology office the other day. Well, why do you even bother to come to lecture if it's all online and everything else other than the stick of the clicker questions in the first couple of minutes? So that's the point. Hopefully, that's why you're here and I'm here, is to do that. So otherwise, again, you just listen to the video. You don't get quite as many hand-waving actions, but other than that. Quick also comment on clickers. Again, I posted all the clicker numbers that were registered from Friday. That's on D2L. Take a look at that list if you're worried about whether your clicker is being registered. I will try this week, hopefully, but certainly by the midterm, to connect those clicker numbers, which are there, to your names. And then if there are questions about that, again, I'll be posting clicker scores after the exam, at least, where we are so far. Any questions about the clickers and such so far? No. OK. Speaking of clickers, it would be that time. So first question for today is, in centromere-specific nucleosomes, which histone is exchanged? H1, H2A, H2B, H3, or H4? Again, feel free to ask your neighbors what they think. They may be wrong, but feel free to ask them. Ten, 5, it's amazing the number of people who vote in the very last second. You can see up the top there how many of them are doing that. So guess what people think? Most of you like H3, so do I. What is the alternative histone that's specific for centromeres? SenPA, which is a replacement for histone H3. Why is that important in terms of understanding how nucleosomes are put together? It's not the question I asked for the clickers. Yeah?
H3 and H4, they form a subunit first. So if they come apart, that means that it changes the way the whole nucleosome is formed if you have a different H3 or H4.
Yeah, so the comment here, so I'm going to repeat that. I should have given you the mic, but I won't do that. I won't put you on display as we have up here. Yeah, that was exactly correct. So what is happening is that H3 and H4 form this very stable tetrameric complex. They're 2 H3s and 2 H4s. And those stay associated with the DNA. And those are the ones that the H2A and H2B exchange a lot more. So H3 and H4 stay associated with the DNA. And it's probably one of the reasons why you have so many modifications on histone H3 on the tails, but also that's the one that gets exchanged. And what that means when you do replication, so we're going to be talking about for the rest of today, is that those stay associated. And so that's going to say, OK, here is the centromere. And once it's here, it's going to stay here. Same is true for those heterochromatin modifications and the euchromatin modifications. So H3 and H4 stay together, and they're much more closely associated with the nucleosome than H2A and H2B. So let me select the appropriate answer, which is correct here. And our next clicker question is, pardon? I'll try and do that as this is going on. So about how much of the human genome encodes unique protein sequences? 1.5%, 5, 10, 50, 75. See, I'll work on the lights here, too. Is that better? Yeah.
Is the easy answer the right one this time?
The right answer is the right answer.
Good. Great.
Been doing this for years. Five. Okay, there seems to be a tiny little bit of confusion here, and that has to do with how much of the genome encodes unique protein sequences. Some people think 50. That's probably because you're getting a little confused with all the repeated sequences which are present in the genome, some of which are clearly viral. That's the about 10%, which are clearly endogenous retroviruses, which, of course, are the most important by far, again, from my totally biased point of view. But in terms of actual unique protein sequence, it's only about 1.5% of our genome, which is really kind of surprising if you think about it. Now, why do we have so little of our genome that's actually making so much of us? And the answer to that is evolution is messy. And that is the best answer I've heard so far as that's concerned, is that it's definitely a messy process. So today we're going to talk about replication. But the sort of key to this is we're not going to be talking anymore about DNA and all of those aspects of things. So I wanted to just quickly go over some of these things. And if people have questions, this was mentioned before, now's the time to bug me, or again, on email, office hours, or next Monday when we have lecture. So 30 nanometer filament, that's where you've got the nucleosomes that are being packed together. And at least in my opinion, most of euchromatin is that way. It's not much more completely compacted. Loops, these are the separate parts of your DNA, which are then going to be separately regulated, separated from other parts of your genome by usually barrier sequences and usually physically attached to other proteins. Talked a bit about topology. I've got my left and right-handed helices here. I posted a couple of links on D2L to some YouTube videos and some descriptions of topology. If you want to take a look at that, some of the stuff there is really nice with a lot of strings and how you can look at strings and look at topology from that point of view. Talk a little bit about epigenetics again, having to do with these alternative histones and how those are changing, also in terms of the modifications that you have on the side chains of usually the tails, usually also N-terminal tails, although to some extent C-terminal tails of the histones. Heterochromatin, this is that extra compacted, and extra compacted means that you can't get other proteins into that complex. This is your silenced DNA, and very often that's silenced in a non-sequence specific fashion, and then is not inherited from generation to generation. So this is the epigenetic inheritance. Again, the modifications of the histones, these are mostly covalent modifications. The most common ones are methylation and acetylation, but you have lots of other modifications as well. Phosphorylation, ubiquitination, sumylation, which we didn't really even talk about, and probably even more than that as well. Alternative histones, we just had a clicker question about that. A little about localization. That's just where any individual chromosome happens to be in the nucleus, and each of them has their own sort of areas in the nucleus where they are. It's different from nucleus to nucleus, but each of them separate from each other, and that's the most important thing there. And then when you have parts of each of those chromosomes that are active, being actively usually transcribed, those are going to be moving away a little bit from their normally compacted regions, and this gives you a pretty good indication that a lot of the business machinery, i.e. the transcriptional apparatus, and also for that matter the replication apparatus, is in a specific place in the nucleus. A lot of people think about these things sort of floating around or zooming along the DNA. That's not how it happens. The DNA comes to the proteins, and that'll become a lot more obvious as we move on through replication today. And then metaphase compaction, it's basically ATP-driven, these little clips that sort of clip the various loops of DNA together, and then eventually the two sister chromatids together. Is there questions on the DNA structure at this point? Okay, let's move on and talk about DNA replication. Again, just quick reminder, a lot of these review slides are probably also a really good way to think about reviewing for the exam. It's partly why I put them in there. DNA replication, we'll talk a little bit about mutations, and these are the changes that happen in DNA that usually are bad, but you have to have them in terms of having evolution be able to take place. Semiconservative replication, hopefully this is not news to any of you, but we'll go through a little bit more detail about how people came to that, a little bit more than they have in the textbook, because I think it's really cool, but I think it's also really good in terms of understanding how some of these mechanisms came to be understood. Talk a little bit about fidelity. Fidelity here just means, yes, you copy most of your genome, including that 98.5% that's not obviously protein coding, really, really well. And how that happens, a lot of it has to do with proofreading, a lot of it has to do with the activity of the DNA polymerases, and to some extent about repair. But mostly it has to do with this amazing machine, which is what's copying our DNA, and it turns out copies almost all DNA for all things. In the back, yes? So yeah, this is sort of a question about really synthetic biology. And so can you cut out the junk DNA, the non-coding DNA, and still get it to function? To some extent, we're lucky that we've been around for, well, at least we in terms of life, probably around for about 4 billion years. A lot of that's been done already. There's a nice discussion in the textbook about the fugu fish genome. Fugu is vertebrate like us, very similar protein coding sequences, missing a lot of that extra stuff. And okay, fine, we're not pufferfishes, but we do seem to work reasonably well, and so does the fugu. That's one way that those kinds of experiments have been done. The other question is about in some of the bacteria, and this is probably more relevant to what's really going on right now. That's cutting out as much of a bacterial genome as you can and still have that be able to function as a bacteria. There's a company called Synthetic Genomics down in San Diego, which has done this with mycoplasma, and they basically synthesized the whole genome of a bacterium, and it still works. It still seems to function and make more bacteria. So those are the ways that people are looking into that. Although it turns out, as I mentioned right at the end of last lecture, kind of got through it really fast at the end, but if you look at what parts of the human genome that rest, again, of that 98.5%, a lot of it's being transcribed. And a lot of it probably is important for what makes the two of us different from each other and everyone else, and for that matter, humans different from chimpanzees, etc. And we don't understand what that is at this point. So giving us a lot of stuff to do. So that replication, again, mutations, relatively rare, but still you have some, and fortunately we've had a really long time through history to look at that. We'll look at the actual process. I mentioned this before when I talked about localization of chromosomes inside the nucleus. That replication fork is sort of the machinery. That's what's doing the job of making more of the DNA, semiconservative replication. Talk about some of the things that go with that. The so-called sliding clamp is a really critical part of that whole replication complex, because it turns out that DNA polymerase is a really big problem. And that is that DNA polymerases fall off the DNA all the time unless you help them stay on there. So that's what the sliding clamp's about. The replosome is just sort of everything together. And then if we get to mismatch repair, supercoiling, this is really the overview for both this lecture and next lecture. So I want to talk a little bit about mutations. All that a mutation is a change in the DNA, and particularly it changes that DNA sequence. And we already talked about base pairing already. Anything that changes base pairing can give you a change in a particular nucleotide, and as long as that gets replicated, passed along, that will be generating a change in the DNA, which is then persisting. Turns out it happens really, really rarely, and it happens with about the same frequency in E. coli and in humans, on the order of 1 in a billion replicated nucleotides. And people argue about this all the time, how common exactly. And so those exact numbers are not absolutely critical, which means I shouldn't be putting them on the exam unless they're really big sort of differences between each of those different numbers. What that means in terms of the human genome is about every time that one particular cell divides, you'll have 3 changes that happen. Because our genome is about 3 billion base pairs in size. Just think about the haploid genome. How many cells are there in a human body? Yeah, there are hundreds of millions of cells in the human body. So there are lots of changes that happen. And that is something to really bear in mind. You think about it, all cells are not going to be genetically identical to each other at the nucleotide level. And this does become important, particularly if you start thinking about cancer and so on and so forth a little bit later on. Because of this mutation frequency, which is just due to replication and immediate DNA repair, it means about the maximum number of proteins you can have, if you do some back-of-the-envelope calculations, between 30 and 50,000 actual proteins. And so the original predictions, in fact, when people were doing the first human genome project, they said we'll have 100,000 different proteins. And then they said we'll have 50,000 different proteins. And then we'll have 30,000 different proteins. And now we're about 20,000 different proteins. So each time when they look at the genome, actually it's getting smaller and smaller. But if you just think about the mutation rates, about the maximum that you can have is around 30 to 50,000. Most mutations are eliminated. Certainly if you think about mutations that are in the germline, those get passed along from generation to generation. They're deleterious, causes something to not function, and they're lost because that particular organism, that particular cell just doesn't make it. Particularly, this is true in cancer. We know that cancer is a genetic disease because of multiple changes that happen. Now, it turns out they're probably not individual point mutations for most of the cases, although there are a couple of cases, in fact, of cancers where it's a single nucleotide change that seems to cause the cancerous development. Few mutations, obviously, are really important because if we hadn't had these kinds of mutations, all of us would be little slime still floating around on the surface of probably the oceans or somewhere in the oceans. So without some mutations, we wouldn't really exist. How does DNA replicate? Again, this is not terribly surprising, shouldn't be new to any of you, but the idea here is that you hang on to the 2 strands that you started with in your double-stranded DNA from Watson and Crick's model. Once those get separated, you copy each one of them. So you end up with 1 old strand and 1 new strand, and this was immediately predicted by Watson and Crick in their paper, which is one of the reasons that people thought their model was really, really cool. There were also people who thought it was totally crazy because how do you pull the 2 strands apart? We'll talk about how that happens in just a second. Turns out it's an incredibly inefficient thing to do. It'd be much more efficient if the information were sticking out on the outsides. But they come apart, those two strands come apart, and then you copy in the opposite direction for each of those strands, because the model only works if you have anti-parallel strands. So if you pull the two strands apart, you end up with this now is the red label is for the old strands, the blue labels are for the new strands. Here, 3' to 5', and opposite here, 5' to 3'. And we'll get back to why this is problematic. Hopefully all of you are going, oh, Okazaki fragments, lagging strands, etc. But we'll get back and talk a little bit more about that later on. So this was the prediction of Watson and Crick. What year was the Nature paper that this came out in? 53. So a lot of people tried to show whether this was in fact correct. And the way that they tried to do this was just looking at what this prediction was. As I already mentioned, in the first generation, you should now have 2 black strands here. Sorry, over here. This is, well, God, this projector doesn't give colors very well, does it? Sort of here, black and orange. Here, the orange are supposed to be the old ones, and the black ones are supposed to be the new ones, or green ones. And you always end up with these one old strand, and then all of this new DNA as you pass through the generations. So in the first generation, you've got 2 DNA molecules, which are half old and new. Next one, 2, which are still half and half, and then 2 that are both new, and so on and so forth through the different generations. That was only one possibility in terms of how DNA could be being replicated. And it turns out that there was a lot of discussion at the time about how this was really happening. And a lot of it had to do with that problem of how do you pull these two strands apart? They're clearly heavily wrapped around each other. How do you get those two strands apart? It might actually make sense to leave those strands together and then just make a copy of that, which a lot of people thought was probably the case. Because again, you don't have to pull the two strands apart. We know in the major groove you have all of the information that you need just by looking at those chemical changes which are there, all the chemical molecules which are there. So what you could have is a case where here's what you start out with, then you make a copy, and then you make a copy of each of those. Of course, the big difference here is that now we have, instead of a mixture of old and new strands in the first generation, we've got one old and one new, and then as you move along, you always hang on to this one copy, and then all of these other ones are different. Now, it turns out there are a couple of cases, particularly in some of the viruses, because they're so cool, which do replicate in this kind of fashion. Probably more importantly is this idea of dispersive replication. And what this means is that there are different parts of each of those DNA molecules that get replicated. And so you end up with mixtures. So you mix and match, a piece here, a piece there. And you end up with half of one molecule and half the other molecule. But you don't pull those two strands apart completely and then make copies of each of those. And this, as it turns out, is exactly what happens in DNA repair, which is what we'll be talking about mostly after the midterm, is it's bits and pieces that get copied each time. And the prediction that you would have here is that after the first generation, you have half and half, because you've got half new and half old. After the next generation, now you're going to have 3/4 which are new and 1/4 which is old. And so you've basically got these 3 possibilities on how this could be happening. So how was the experiment done? Hopefully, again, none of this is new to you. The Michelson-Stahl experiment. This was done by Matt Michelson and Frank Stahl. Frank Stahl, emeritus professor down at U of O. I think he's still maybe teaching. He's certainly around and very active down there. He came up and gave a talk a couple of years ago. But really fascinating guy. But he and Matt Michelson came up with this really nice experiment whereby they took E. coli and grew it, and this was really Frank Stahl's contribution to this. growing E. coli with very specific nucleotides that had a heavy precursor to them, and heavy was the N15. And the reason that they could actually do this experiment after the Manhattan Project, nobody knew what to do with all their isotopes and all of their various different excuse me, heavy nucleotides, et cetera. So it was sort of an offshoot of that, which allowed them to even do some of these experiments. They weren't, in fact, radioactive, N15 versus N14, but they were generated through this process. And then after you'd grown the E. coli with your N15, then you switch it to N14, a regular isotope of nitrogen, and then separate the DNA. And then most important in terms of this experiment was the development of this, also known as the big machine, which is going to be on the next slide, equilibrium analytical ultracentrifugation. And so basically taking your sample, spinning it really, really fast, and then allowing everything to separate in terms of the density. And it turns out that's what Matt Mieselson's contribution was to this whole experiment, was to figure out the process of how you could get separation based on the density of what you were looking at. So one of the reasons that this was such a cool experiment, according to me and other people as well, is there were really clear predictions about what was going to happen. You start out with your heavy DNA, you make a switch, and then you have three different possibilities. After one generation, it can be half and half in semiconservative or in dispersive replication, because just half and half. However, if you have conservative replication, you're going to still have those two strands which are old, and then two strands which are new. And then after the next generation, in semiconservative, you'll have some which are all brand new, and still some which are half and half. And that differentiates from your dispersive replication here. How do you do this? You use the big machine. A little hard to see over here, but this is Matt Mieselson next to the Model E analytical ultracentrifuge. It's literally like a couple of fridges right next to each other. These are really big machines. And this rotor here is what he's spinning at about 60,000 RPM overnight with a little window in it, and that's right here, where you can shine UV light through it and then detect where any of your different pieces are looking at that. So the results are here. One thing I wanted to point out here is that it was only 5 years after Watson and Crick had published their model. And what you see, and they don't have exact numbers here, but you start out with your heavy DNA. Here now is your light DNA. You see there's not very much difference between these things. They're actually pretty darn similar to each other, and even the analytical ultracentrifuge had a hard time separating from each other. So after one generation, now you have just a single band, which is that half and half. This could now be dispersive or semiconservative. So it's only after 2 generations, or at this point 1.9 generations, we now have your half and half, and then your completely new. So this was the experiment which pretty clearly showed that you had semiconservative replication. What's interesting, in their paper, they actually just show blocks. They don't show the different nice strands wrapped around each other, because there was still this problem of how you pull the strands apart and how you put them back together. And that leads to the enzymes which are actually doing the job. And this is really the work of Arthur Kornberg, also known as Kornberg Sr. I think one of the very few cases where father and son have gotten Nobel Prizes. Arthur Kornberg discovered this enzyme, also known as DNA polymerase I, even before the publication of semiconservative replication. So they knew that DNA polymerase was a really important thing. And again, they've been working on it since Watson and Crick had come out with this particular structure. So Arthur Kornberg and his students discovered this enzyme, which did one particular activity, and that was it would take nucleotide triphosphates, in this case deoxynucleotide triphosphates, and add them to a primer as long as there was a template around. So the template That's what you're going to be copying. The primer gives you a 3'OH. And it's that 3'OH which is really, really critical. You have to have that 3'OH. The polymerase adds a nucleotide to that 3'OH. That process then gives you, goes from a triphosphate to a monophosphate, leaves you another 3'OH. So the process can go again and again and again. What's the chemistry? It's the chemistry we talked about many times before. It's all about the water. What's that reaction called? It's a hydrolysis reaction. You're hydrolyzing the gamma and beta phosphates from your nucleotide triphosphate. This is what's called pyrophosphate that's removed, and then you have a secondary hydrolysis that takes your pyrophosphate and generates the monophosphates. It's that coupled reaction that, again, we talked about right in the, I think, second lecture, which gives you a real directionality to this process. You don't want to be depolymerizing your DNA all the time. So you want to be going through and adding each of these nucleotides. And again, this is to hopefully beating on the dead horse here. You're going from a 5' end to a 3' end. You're always adding on a 3' end. So your polymerase is going to be polymerizing from 5' to 3'. And then the template strand is always going to be opposite. So it's 5' to 3' in this, in this model. The structure of DNA polymerase 1 kind of looks like a hand. Always people talk about the hand process. Here, the thumb, fingers, and palm. Basically, the business part of the DNA polymerase is in the palm. That's where you have the polymerization that takes place. But basically, this hand really kind of grips the DNA. And it's a little bit hard to see down here at the bottom. But that's the palm. Here are the fingers. There's the thumb. And basically, it's this gripping process that's your first step in fidelity, that's telling you you've got the right base pairs that have formed. And so that base pair formation is really critical. And as you remember from Watson and Crick's structure, A's are going to pair with T's and G's are going to pair with C's, giving you an identical or almost identical structure of the backbone of your DNA. So the backbone of your DNA doesn't change. Where those sugars are is identical if you have a GC or an AT. If you have something different, that's going to be slightly different. And that turns out is the first step in terms of your specificity. The other thing that was only figured out once they had a high-resolution structure here is that your template, and that's that extra strand that you're adding to, is slightly bent. And it turns out that slight bending gives you that possibility to check exactly that you've got the right base coming into the right place on your template. And that's actually before the ligation takes place. And if you think about it, that kind of makes sense. You don't want to have these things right next to each other. You're not going to have the incoming nucleotide. You want to be able to check it before you have the chemistry that takes place and does that connection. Yeah?
Is that kink in both strands, or is it after they've been?
Okay, so the question is the kink in both strands or only afterwards? It's only the template strand. It's when you're adding the next nucleotide. And so as soon as you've added that, then it becomes a straight piece of DNA. So this is great. It's wonderful. But this is only going from 5' to 3'. And we talked about semiconservative replication. These guys only work when they're opposite orientations to each other, right? So how do you go about replicating that other strand? And apparently, Arthur Kornberg was not known as being the most tactful researcher, was trying to get his students to find that darn 3' to 5' polymerase. And they couldn't find it, couldn't find it, couldn't find it, couldn't find it, couldn't find it. Still got the Nobel Prize. But in the meantime, they showed that, in fact, you had circular pieces of DNA, and they would replicate, and they were clearly replicating from 5' to 3' and 3' to 5'. So what's going on there? Okay, hopefully all of you know. It has to do with the Okazaki fragments. Reiji Okazaki and his wife, Seiju Okazaki, found that there were these short pieces of DNA that were being made at the same time as the long piece of DNA during this replication process and then stitched back together. This is why they also now call them Okazaki fragments. Turns out that Okazaki fragments in bacteria, which is where they were originally found, and also in bacterial viruses, go viruses, are about 1,000 bases in length, whereas for eukaryotes, they're much, much, much shorter. And it turns out the technique that they were using to detect these only was able to detect those longer ones. The shorter ones, they weren't able to detect at all, which is a nice plug for the other course I'm teaching next term, which is called Molecular Genetics, where actually we'll read, among other things, the original paper where the Okazakis showed that this was what was going on there and talk about that process. So again, turns out that there's multiple separate pieces, and they, in fact, originally thought that both strands were being made this way in separate little pieces relative to each other. So I mentioned here it's all about the polymerase and how you get that first step of fidelity. How do you get the correct nucleotide?' that template is kinked, which allows the nucleotide to come in properly and before it's made the covalent linkage to the 3' OH. But the detection process actually turns out to be much more about not the actual hydrogen bonds, because hydrogen bonds are going to be binding to the other base. They're not going to be binding to the protein. So how do you know that you have that right base pair which is formed? Well, it really has to do with the fact that the sugars are in the right place. And they mention in the book that there's actually sort of a grab. The hand makes a little grab. And if it grabs properly and you've got a nice flat base pair with the sugars in exactly the right place relative to each other, then you will incorporate that nucleotide. And it's really much more about the stacking and the base pair itself. It's not about the individual nucleotide. So it's really checking that geometry to make sure that everything is correct. If you don't have that correct base pairing interaction, then what will happen is things get stuck, and you're not going to incorporate that base. Or if you do, you go to proofreading, which is the next step. Yeah, question?
You mean stacking of the sugar phosphate backbone?
Okay, so the stacking question is stacking of the sugar phosphate backbone. No, what I'm talking about here is the stacking of the bases on top of each other. And so it's the base pair stacking. And you remember we talked about this before. If you look at how base pairs are in the middle, they're really stacked down on top of each other. And a lot of the stability you get from DNA is due to those electron interactions in all of those pi rings which are present in the bases. Right in the back, yes? Yes, you're getting way ahead of yourself here, or all the rest of us here, stuttering. Stuttering actually is usually not thought to be the case for replication, much more so for transcription. And when we get to transcription, we'll talk a lot more about stuttering when we get there. Turns out regulation of extension mostly has to do with stuttering there. And then for replication of virus genomes, but that's next term too. So yeah, no, for the DNA polymerases, they actually seem to go relatively consistently. With, again, these changes when the incorrect nucleotide is incorporated there. Yeah?
Okay, he went forward. I think I need to go back.
Yeah.
I just wanted to be sure something really basic, which is that is the DNA polymerase responsible also for that separation of the two strands at the onset?
Okay, so the question is the DNA polymerase responsible for the separation of the two strands at the onset? No. We'll get to that in just a second. Yeah, so the question is that conformational change, is it to test if it's the right pair? And so what this figure is supposed to be showing down here at the bottom is, well, actually the top here really, is an alpha helix, which is part of the DNA polymerase itself. What that does, it basically, they talk in the book, they talk about a hand closing, but really what's happening is it's this alpha helix which has these side chains on it, particularly this tyrosine side chain. Tyrosine, of course, has this aromatic structure to it. That stacks down right on top of the base pair if it's the correct base. And it will fold down flat, so that's that nice conformational change. If you don't have the right base pair, it's a little squiff-wise, you're not going to get that appropriate packing. Okay, so that gives you the first step of fidelity. But that's not a 1 in a billion fidelity. The second way you get fidelity is if that folding down doesn't work properly. And it turns out that these polymerases are going really fast, thousands of nucleotides a second. So occasionally you'll have problems. Also, when we talked about the DNA structure, or maybe when Eric talked about the DNA structure a little bit, you have these tautomers, keto and enol groups in the various nucleotides. Sometimes those will have isomerized. And so you'll have an OH instead of a double bond O, or an NH2 as opposed to an NH. Those will base pair incorrectly. But tautomerization happens, then you end up with a base pair that's not quite right. And again, it's because then you've got the two pieces on either side that are wrong. And it turns out that most, not all, but almost all DNA polymerases have a way of getting rid of that incorrect base. It's what's called proofreading. And basically, again, it's the structure which is wrong. It's not detecting that it's a base pair. It's the structure of that base which is there. And then there's a 3' to 5' exonuclease. So it's, again, talked a little bit about enzymes before. ACE at the end means that it's going to be some kind of enzyme. Usually it's going to be a hydrolytic enzyme. What does it work on? It's a nuclease, so it's breaking down nucleotides. There's also exo versus endonucleases. Exo is cutting in from the ends. Endo is cut in the middle. So this, just the name of it, tells you exactly what it does. It's a 3' to 5' exonuclease. So it chews back what's already been made. And once it's done that chew back one step, it leaves the 3' OH. Now that 3' OH, of course, is exactly what you need to extend from, because that's the chemistry of the polymerase reaction. Yeah?
And that exonuclease is a domain within the replosome?
So the question is the exonuclease domain within the replosome. So that domain, the excuse me, the 3' to 5' exonuclease domain is part of the DNA polymerase itself. So it's not part of the replosome. We'll talk more about the replosome later on. But that exonuclease domain is present in all replicative DNA polymerases. And we'll talk about some of those other ones later on. Yeah? Yep.
The enol form is favored for the action of the polymerase. What keeps it in that since it drives towards the ketone form?
You'll have it's an equilibrium between the two. So the incorrect one happens with a certain frequency, and the correct one, and these will bounce back and forth. There'll be less of them when you have hydrogen bonding, which is formed, because most of them are finding those hydrogen bonds. But once you've actually incorporated it, it doesn't really matter, because that isomerization, you've got the appropriate nucleotide which is there. It's just when the incorrect isomer gets incorporated. And then when that isomerizes back, then you need to get rid of it. And that's what this is all about, this proofreading prospect. Yeah?
So this is happening at the same time as replication, just by different domain on DNA polymerase?
Exactly. And we'll take a look at where that is in just a second. So sorry, the question is this happening at the same time as polymerization? Yes, polymerization and proofreading are happening simultaneously with each other. So that process is happening all the time. So one thing I did want to mention with this, and they spend a while in the textbook talking about this, this proofreading only works if you have 5' to 3' polymerization with nucleotide triphosphates at your 5' end. And the reason for that is basically shown here. Unfortunately, this is a figure from the 5th edition of the textbook, not the 6th edition. I don't know why they took it out. But here, the normal process is, again, you extend from the 3' end, you have your triphosphate, lose your pyrophosphate, that gets extended. If it's the incorrect nucleotide which gets added, you have your exonuclease, which comes in, chops off this last added nucleotide, and you can continue just as it's going along here. If, however, on the other hand, you are extending from 3' to 5', so the 3' end over here, 5' end over here, attaching on the 5' end, here you have addition. You add this nucleotide triphosphate, that's no problem. But now if you cleave off this last nucleotide, because it's the wrong one, it was the isomer which got added incorrectly, now you end up with a 5' monophosphate. You can't add to that anymore. There's no pyrophosphate which can be removed. So the fact that you have 5' triphosphates that you use for incorporation, they have to add to the 3' end if you can have efficient proofreading, because it always generates that 3' OH. Or it would generate a 5' phosphate, but that 5' phosphate is no good. You can't extend from it anymore. So this is probably why, evolutionarily, we only have the 5' to 3' polymerization activities because of that proofreading activity. So what does that proofreading activity do? And this is getting back to your question. Is it happening at the same time? Here's your hand polymerase. I much prefer the hand polymerase version. There's a little clamp thing in the textbook. Horrible. The hand's way better. Going through polymerization. Again, the business end of your polymerase is here, down in the palm of the hand. This is where you have that kinked template. You're checking here, make sure this has got the right base stacking. If it is, you continue. However, if now this base pair is incorrect, this base gets flopped to a different site, a different domain in the polymerase, and that then gets cut off. Between that first step, checking the base, that it's the correct base, and proofreading, what you end up with is about a 1 in 10 to the 7 mistakes, which is still not your 1 in a excuse me, billion mistakes for replication, but it's pretty darn good. The last step is what's called this mismatch repair, strand-directed mismatch repair, which at this rate we're probably going to get to on Friday. And together, that gives you between 1 in 10 to the 9 and 1 to the 10 to the 10. And as I mentioned when we did that first slide, people argue about which one of these is exactly right. Again, depends on which data you actually look at there. So that's the polymerase, and that's its activity. It's got this normal 5' to 3' polymerase activity, the 3' to 5' exonuclease activity. But it has this problem, and that is it can only add on 3' OHs, which is great and wonderful and well and good, except you got to start somehow, right, when you pull those two strands apart. So how do you do that? You have to have not a DNA polymerase, but an RNA polymerase. And that RNA polymerase is also called DNA primase because it's making the primer. So it's a bit of an exception to that rule that I gave you. Aces are always breaking things down. Here it's actually making something. So this DNA primase, which is represented by this blue blob right here, is an RNA polymerase. And that RNA polymerase, like all RNA polymerases, is actually relatively low fidelity. It doesn't have much proofreading activity, and it turns out there's a tiny little bit of proofreading activity in RNA polymerases they don't talk about in the textbook. Again, it's biology. There are always exceptions. But for the most part, relatively low fidelity, certainly as compared to the DNA polymerases. It generates your primers. And once, of course, you've made a primer, now you have a template here and a 3' OH. So your DNA polymerase can extend that. And here's just an example of that right here. Here's your RNA primer. Your DNA polymerase is extending from 5' to 3'. You also have another primer, and certainly for the lagging strand, you have to keep doing this. Another primer, 5' to 3', extends, etc. Well, once this 3' end here ends up running against one of these primers, you need to get rid of this primer. And there's a couple of ways of doing that. One of those is actually a DNA polymerase itself. In fact, the first DNA polymerase to be found is DNA polymerase 1, which we'll look at in just a second. And there's also something called RNase H. Now, RNase, what does it do? Chops up RNAs, excuse me. H stands for what? RNase hybrid. H stands for hybrid. And hybrid is just DNA-RNA duplexes. So all that RNase H does is it chops up RNA, which is in an RNA-DNA hybrid, which is great. And then once you've done that, now your DNA polymerase can keep going. You end up with a gap between the 3' OH of the nucleotide, which you've just added, and the 5' phosphate that was there that you've either chewed back to, usually in the case of RNase H. Those need to be hooked together. There's a protein called DNA ligase, which will hook those two together. This is just shown in a slightly different way. This is now the figure from the Watson textbook. Here we have your primer. Your RNase H will chop this out. Now you've got a 3' OH and a template. This gets extended. You end up with this little hole right here. This then gets hooked up to give you a continuous strand. Did want to mention here this DNA polymerase 1. DNA polymerase 1, again, it's the first DNA polymerase to be found by Roger Kornberg's lab. It had this wonderful 5' to 3' DNA polymerase activity. That's how they found it in the first place. It had this proofreading activity, the 3' to 5' exonuclease, but it also had a 5' to 3' exonuclease activity, which was really bizarre because, 5' to 3' is the same direction that you're polymerizing. If you also have an exonuclease activity, that's going to be chewing up whatever you've already made. So at first, they tried to purify these things away from each other. They tried to purify the two things away from each other. It's only later that they found that that's exactly what you need to get rid of primers with. Again, they didn't know about primers at the time. So DNA polymerase 1 has 5' to 3' exonuclease activity and 3' to 5' exonuclease activity. So it's got both. So it chews in ahead of itself and will also back up. So you've got a proofreading activity and a primer removal activity in DNA polymerase 1. Turns out DNA polymerase 1 is specific to bacteria, or particularly actually E. coli. You don't find this activity in eukaryotes, and we'll talk about a different activity that's important for that. Yeah, question? Yeah. Okay, the question, I'm not sure I quite understand your question here. Yeah. Mm-hmm. Okay, so the question is, how does the DNA primase and the RNA polymerase relate to each other? So the DNA primase is an RNA polymerase. So it's providing the primer for DNA polymerization, but it's an RNA polymerase. You can also, and people will often talk about these as, what the template is that they use. And so in this particular case, the DNA primase is a DNA-dependent, so that's the template, RNA polymerase, because that's what it makes. If you think about DNA polymerases, those would be DNA-dependent DNA polymerases. Yeah? Yeah.
On the second figure, does the RNase H not get rid of all of the RNA primer?
So the question here is basically, what about this little green thing that's left here? It turns out that, so the question is that RNase H, and RNase H doesn't do the whole thing. It varies, and this may even be a typo in the textbook. Sometimes RNase H will take all of it out. Sometimes it'll leave one. But particularly if you've got one of these DNA polymerases like DNA polymerase 1 that has a 5' to 3' exonuclease, it can get rid of it. Whereas if you have a polymerase that doesn't have that activity, and that's actually most of the eukaryotic nucleases don't have that. Eukaryotic polymerases don't have that. It won't be able to remove that. So they're much more dependent on RNase H, and then we'll get to some endonucleases next time. Yeah?
Is there a higher margin of error on the lagging strand because there's more stuff going on?
So the question is there a higher margin of error on the lagging strand because more stuff is going on? And basically, I'll rephrase your question and say, do you have more mutations that happen on the lagging strand than you do on the leading strand? That's actually a great question, And a lot of people are really interested in trying to figure that out. At first, there was some evidence that, yes, that was the case. Now that evidence seems to be being reversed, and we may get to that, again, probably on Friday at this rate, when we talk about how leading and lagging strands are being made. But it doesn't seem there's a big difference between the two. And that's actually a little curious because, again, you've got these low fidelity polymerases. But it seems that getting rid of those RNA primers is so efficient that you don't have too much of a problem there. And we'll see how that happens again in the repliposome in just a second. Yeah?
Places where DNA primase attaches, is it random or are there certain sequences that are targeted?
Come back on Friday. No, so yeah, the question is where does DNA primase know where to go, basically? And that's the origins of replication, and we'll talk about that on Friday. Yeah?
So you said that the DNA primase is an RNA polymerase, and you said what it does.
Okay, so basically DNA primase is an RNA polymerase, and it makes the primers for your DNA polymerization. So that's what it does. And in terms of those primers are all RNA. Okay, let's get to the next problem. And the next problem is how do you get those two strands apart in order to have DNA primase, which is going to come down and bind at the origins of replication, but also as you are replicating. As you're replicating, you're pulling the two strands apart because the only way that your DNA polymerase can be incorporating that next nucleotide is to have the template actually be free. So how do you get these templates separated from each other? This is our friend, another ACE. What does it do? It works on whatever comes in front of that. That's the helicase. And so what does it do? It breaks helices. It gets onto one strand of DNA and pulls it apart relative to the other strands. Turns out that this is an incredibly ATP-dependent reaction because two strands, wrapped around each other, pretty stable. So probably one of the main things that's burning ATP during replication is the helicase, is pulling those two strands apart. So the people who thought right at the beginning that Watson Crick's idea of the two strands coming apart and then copied, it is tough to do. But that's what these helicases do. And again, they're circles, wrap around a single strand of DNA, and just crank right along it here. And I like this figure down here in the corner because it gives you an idea of the size of the individual proteins relative to the DNA. So DNA is pretty small relative to this. And the way that these work, and I've seen some videos, but I don't actually have a nice example of this, but they literally spin around these helicases, and in that process are just pumping one single strand down the middle. Down the middle of this structure is only enough space for a single strand. There's not enough space for two there, so it has to be a single strand. And so it's basically pulling the single strand through the helicase and giving it that process. Once you've made a single strand, this single strand is actually really quite unstable. What it would love to do is bind to an opposite strand. In fact, we know that's what DNA loves to do. RNA is the same way. Loves to form double strands. So forming those double-stranded structures would happen if you didn't have one of these single-stranded binding proteins. That's what SSB stands for. It's just single-stranded binding protein. This is the SSB that you find in eukaryotic cells. It's also called RPA. And then here's the structure. Again, not dissimilar to the case with the helicase. It only binds to single strands. And since it only binds to single strands, that's going to be protecting that single strand from binding back to other single strands. But also, it turns out single-stranded DNA is not very stable, just chemically. And so it protects it from any kind of chemical damage. The other thing that I needed to mention here is that this single-stranded DNA binding protein is highly cooperative. What do I mean by highly cooperative? Yeah, it likes its neighbor. But no, it likes its neighbor so much that once one of them is bound to DNA, getting a lot more of them bound to DNA at the same time is much, much easier than that first step that happens on that very first interaction of the single-stranded DNA binding protein with single-stranded DNA. So your helicase pulls the two strands apart, single-stranded DNA binding protein is going to bind to that. And it turns out that both of these are absolutely critical for getting any kind of replication, and people found these out by classic genetics, make mutations, find things that have problems, figure out what they do. And so RPA is replication protein A, and then the helicases are also found. These are all mutations originally found in E. coli that had problems with replication, and so eventually found out what they did. So helicase pulls the two strands apart, single-stranded DNA binding protein binds to that, and then, of course, the polymerase needs to come in and replicate this piece of DNA, and the DNA polymerase can actually bump off each of these single-stranded binding proteins because that interaction is actually stronger than the interaction of the single-stranded DNA binding protein to DNA. DNA polymerase, however, has a problem. And that problem, as I kind of mentioned before, is DNA polymerases fall off the DNA all the time. And so they're also known as non-processive enzymes. They'll make a couple of nucleotides and then fall off the DNA. They'll make a couple of nucleotides, fall off the DNA. Well, if you've got to go through thousands of nucleotides a second, if you think about the E. coli genome, it's 5 million nucleotides long, replicates in about 30 minutes. You've got some cranking polymerase along here. And if that polymerase is falling off all the time, it's going to be a major problem. So what you need to do is have some way of keeping this DNA polymerase onto where it's supposed to be. And that way of doing that is called the sliding clamp. This is another circular protein that goes around the DNA. But unlike the helicase that just goes around a single strand, these guys go around double strands. And they're also known as processivity factors because they make the DNA polymerase stay on the DNA or stay where it's supposed to be. So once you have one of these processivity factors, that now interacts with the DNA polymerase, keeps it on the DNA, and allows it to polymerize, in the case of E. coli genome, starting at one position, the bidirectional replication origin, again, we'll talk about this more later, goes 2.5 million bases without falling off. And that's because of this interaction with the sliding clamp. Turns out that you have a very similar structure in all different organisms, but they're put together in very different ways. So it turns out the sliding clamp that you have in bacteria is made up of two proteins. It's a dimeric protein, whereas in the case of eukaryotic cells, it's called pCNA, absolutely horrible name, but it's proliferating cell nuclear antigen. Okay, why do you know about proliferating cell nuclear antigens? Well, what do proliferating cells do? They replicate. They are replicating on leading and particularly on lagging strands. So what do you need a lot of? You need a lot of these sliding clamps. So it's actually not terribly surprising that proliferating cells have a lot of this particular protein. So pCNA, I think, makes a lot more sense. Last couple of minutes of class, I want to talk about the obvious problem that should have been coming up in all of your heads, which is, how the heck do you put a ring around DNA? And the answer to that is you have a clamp loader, which takes this pCNA or the beta protein that you have in bacteria and holds it in open position till it gets onto the DNA and lets it close. And that's all that this guy does. It's ATP dependent because the stable position that you've got for any of these sliding clamps is circular around the DNA. That ATP hydrolysis will allow the clamp to close down on DNA. Yes? Pardon? Okay, so the question is, do you need one of these clamp loaders for single-stranded DNA as well as for double-stranded DNA? The answer is not for the sliding clamp, but you do need one for helicases. So you need helicase loaders as well. You're exactly right. The same problem. If you've got a circle going around, how do you get it around there in the first place. So this is just basically what I've said already. Sliding clamps hold the polymerase on. It tries to fall off, but because it's bound to the sliding clamp, it stays associated with the DNA and continues to replicate. We'll look at the replication fork and the replisome on Friday.